Today we will discuss types of enzyme inhibitions. So before going into the types of enzyme inhibitions, first we will see what do you mean by enzymes. So enzymes are the substances that increase the rate of a reaction. The reactants binds to enzyme and as a result products are released. Enzymes accelerate reactions as much as 10 power 16 overall uncatalyzed reaction. So in presence of enzyme, a reaction will be fastened by 10 power 16 times. Next feature of enzyme is specificity of enzyme towards its substrate is controlled by the structure of enzyme. Specificity of an enzyme towards its substrate is controlled by its structure. This unique fit of substrate with enzyme controls the selectivity of substrate for its product formation. This unique binding of substrate with enzyme controls the selectivity of substrate for its product formation. Coming to the topic, that is enzyme inhibition. Inhibitors are the substances that bind to an enzyme and interfere the enzyme activity. The process is called enzyme inhibition and the substances are said to be inhibitors. So inhibitors are the substances that bind to an enzyme and interfere enzyme activity. The inhibitor can prevent the formation of enzyme substrate complex or they can prevent ES breakdown, enzyme substrate complex breakdown to enzyme and product. Inhibitors generally are chemicals that reduce the rate of enzymic reactions. Inhibitors are the chemicals that reduce the rate of chemical reactions. Coming to the representation, enzyme inhibition. Enzyme plus substrate gives rise to enzyme substrate complex. This will forward to give enzyme and product. This condition occurs in absence of inhibitor. So the general enzyme catalyzed reaction can be represented as enzyme plus substrate gives rise to enzyme substrate complex. This enzyme substrate complex leads to the formation of product and releases the free enzyme. In case of enzyme inhibitions, so the enzyme has active sites. To these active sites, generally substrate will come and bind, but in presence of inhibitor, what will happen means there is probability of binding inhibitor to the substrate. So enzyme plus substrate plus I gives rise to enzyme substrate inhibitor complex or EI complex, or simply enzyme inhibitor complex can be produced so that the reaction cannot be forwarded and no product formation takes place. So the inhibitor can bind to the enzyme directly and form EI complex, or it combine with the ES complex and form ESI complex. These inhibitions can be of irreversible or reversible. The enzyme inhibitions are of two types, irreversible inhibitions, reversible inhibitions. In the same way, inhibitors are also two types. They are the irreversible inhibitors and reversible inhibitors. Coming to the enzyme features, enzyme inhibition features. Irreversible inhibitors bind to enzyme through covalent bond. Irreversible inhibitors bind to the enzyme through covalent bond so that they cannot be dissociated from the enzyme and the reaction cannot be forwarded. So these reactions are irreversible reactions and hence the inhibition is termed as irreversible inhibition. Coming to reversible inhibitors, they will bind to the enzyme through non-covalent interactions. Irreversible inhibitors bind to the enzyme through non-covalent interactions which are weak. So they can be dissociated from the enzyme and form free enzyme which can bind to substrate and forward the reaction. Hence, such type of inhibition is called reversible inhibition, and the inhibitors are called irreversible inhibitors. Coming to the types of inhibitions and inhibitors. Let us see reversible enzyme inhibitors. So, inhibitors are the substances which inhibit the enzyme catalysis. So, inhibition of enzyme activity in which the inhibiting molecular entity, that means molecular inhibitor, can associate and dissociate from the enzyme binding site, that is from the active site. So, reversible enzyme inhibitors are the substances which can associate and dissociate from the protein's binding site, that means from the enzyme's active site. There are three types of enzyme inhibitors based on the type of inhibition they are involved. They are the competitive inhibitors, non-competitive inhibitors, and uncompetitive inhibitors. So, enzyme inhibitors are of three types. They are competitive inhibitors, uncompetitive inhibitors, non-competitive inhibitors. In the same way, enzyme inhibitions are of three types. They are the competitive inhibitions, uncompetitive inhibitions, and non-competitive inhibitions. Now we will see in detail about competitive inhibition. Competitive inhibition. Here, these competitive inhibitors, the name itself competitive, these competitive inhibitors compete with the substrate molecules for the enzyme's active site. The inhibitor action is proportional to its concentration. It can reverse inhibition by adding more substrate. That means here the inhibitor competes with the substrate to bind to the active site. So when the inhibitor concentration is more, there is probability of inhibitor to bind to the active site. So this inhibition can be overcome by increasing the substrate concentration. So once the substrate concentration is increased, what will happen? Availability of number of substrate molecules will be increased, and these substrate molecules compete with the inhibitor and will go and bind to the active site as their number is more. Hence, so it is called as competitive inhibition, and the inhibitor is called competitive inhibitor. So what will happen? Let us see in this diagram. So this is the enzyme and this is the active site of the enzyme. So in presence of inhibitor and substrate, both the inhibitor and the substrate will compete for the active site, and which will win will go and bind to the active site of the enzyme. If the inhibitor binds to the active site, the enzyme EI complex will be formed and product cannot be produced. If the substrate binds to the active site by competing the inhibitor, then ES complex will be produced, which leads to product. So let us see an example. For example, succinate dehydrogenase is the enzyme catalyzing succinate to fumarate. This succinate dehydrogenase substrate is succinate, but malonate is a competitive inhibitor which competes for the active site present on the succinate dehydrogenase. As a result, what will happen in presence of malonate? Succinic acid cannot bind to succinate dehydrogenase and fumarate is not produced. Thus, malonate is the competitive inhibitor for succinate dehydrogenase. The second type of inhibition is uncompetitive inhibition. So here a competition is not there. So coming to uncompetitive inhibition. In this mode of inhibition, the inhibitor only binds to the ES complex. The inhibitor binds to the ES complex. I said that enzyme plus substrate gives rise to ES. ES will forward into enzyme and product. But in uncompetitive inhibitor, the inhibitor go and bind to the already formed enzyme substrate complex. As a result, ESI complex will be formed. The binding site for inhibitor is created only upon substrate binding. So why the inhibitor will go and bind to ES complex only means once the substrate is bound to the enzyme, then only the binding site will be generated for the inhibitor in case of uncompetitive inhibitor. So there will be no EI complex formation, only enzyme, ES and ESI, but ESI cannot make product. So only enzyme and ES complexes can be formed. The formed ESI cannot be converted into product. Example for uncompetitive inhibition are tetramethylene sulfoxide and 3-butyl thiolene 1-oxide are uncompetitive inhibitors of liver alcohol dehydrogenase. So what are the examples? Tetramethylene sulfoxide and 3-butyl thiolene 1-oxide are uncompetitive inhibitors of liver alcohol dehydrogenase. So this is a representation for uncompetitive inhibition. I said that only after binding to the substrate, that is after ES complex only, inhibitor will come and bind and form ES complex. So let us see. This is the enzyme, this is the enzyme, this is the substrate. So this is the free enzyme and this is the active site. So enzyme, substrate, E plus S, enzyme, substrate. This is ES complex. Generally in absence of inhibitor, this ES will be converted into free enzyme and product will be produced. But in presence of uncompetitive inhibition, what will happen means after the formation of ES complex, that means once the substrate is bound to the enzyme, ES complex will be formed and then to this complex the inhibitor will come and bind. So here the inhibitor is bound. As a result, ESI complex is produced. So this is the enzyme, substrate is bound to the active site and inhibitor is bound to the another site and this is the ESI complex and this cannot forward to form product. Hence the enzyme reaction is inhibited. Now we will see the third type of inhibition, that is non-competitive inhibition. Here these inhibitors can bind to the enzyme or enzyme substrate complex. In case of uncompetitive, the inhibitor bind to only enzyme substrate complex. But in case of non-competitive inhibition, the inhibitor can bind to the enzyme or ES complex. So enzyme can bind both substrate and inhibitor, but ESI complex cannot make product. This inhibitor inhibits by binding irreversibly to the enzyme, but not at the active site. That means here non-competitive inhibitor do not bind to the active site. They bound to an additional site and the binding is irreversible. So let us see an example. The best example is cyanide. The cyanide combines with the iron in the enzyme cytochrome oxidase. The cyanide combines with the iron in the enzyme cytochrome oxidase. So this is the diagram comparing the non-competitive inhibition and competitive inhibition. So let us see what's happening in non-competitive inhibition. So this is the enzyme, and this is the active site. So the substrate. In non-competitive inhibition, the substrate is not binding, the inhibitor is bound. Non-competitive inhibition change the shape of enzyme so that it cannot bind the substrate. So non-competitive inhibitors will change the shape of the configuration of the active site so that the substrate cannot bound and as a result the inhibitor can bound to the enzyme and form EI complex so that product cannot be produced. Coming to the competitive inhibition, so this is the substrate. In competitive inhibition, there arises a competition between the inhibitor and the substrate so that substrate cannot bind. the inhibitor compete with the substrate and the inhibitor binds to the active site. Competitive inhibition interferes with the active site of enzyme so that substrate cannot bind. That is the main difference. In non-competitive inhibition, the substrate is not binding at the active site. But in competitive inhibition, the inhibitor competes with the substrate so that it cannot bind to the active site, and the inhibitor will bind to the active site. As a result, the reaction is not forwarded. So coming to the second category of inhibitions, those are the irreversible enzyme inhibitions. So irreversible. So in presence, these inhibition reactions cannot be reversed. Coming to the irreversible inhibition. The binding of an irreversible inhibitor can stop a substrate from entering the enzyme's active site from catalyzing its reaction. So the inhibitor will stop a substrate to enter the enzyme's active site and forward the reaction. In case of irreversible inhibitors, they usually react with the enzyme. Irreversible inhibitors react with the enzyme and change the enzyme composition so that they cannot bond to the substrate. Coming to the irreversible enzyme inhibitors or types of enzyme inhibitions. Irreversible enzyme inhibitions. So irreversible enzyme inhibitors bind covalently. They bind covalently. That means strong association between the enzyme and inhibitor, and destroy the functional group of the enzyme that is essential for its activity. That means they will alter the active site configuration so that the substrate cannot bind to the active site and cannot lead to the product formation. These irreversible enzyme inhibitions are of two types. They are competitive inhibition and allosteric inhibition. Competitive inhibition and allosteric inhibition. Now we will see what's happening in the competitive inhibition. So here the permanent binding of the competitive inhibitor to the active site will take place. So this is the actual substrate which has to bind to the enzyme. But in presence of competitive inhibitor, instead of the substrate, the competitive inhibitor will go and bind to the active site, and as a result the substrate cannot bound and the reaction cannot be proceeded. Coming to the allosteric. Allo means different, steric means configuration. Here the inhibitor, besides binding to the active site, it binds at the allo site, different additional site, and those sites where the inhibitor binds is called allosteric site. The permanent binding to the enzyme allosteric site. Here the allosteric inhibitor will permanently bind to the enzyme allosteric site. This allosteric site is a additional site besides the active site of the enzyme. These allosteric inhibitors changes the shape of the enzyme. So this is a representation of the allosteric inhibition. If you observe, this is the enzyme, active site and additional site where the allosteric inhibitor will come and bind, and this site is called allosteric site. This is called active site where the substrate binds. So let us see what's happening in presence of the substrate and inhibitor. So the substrate will come and bind to the active site. Now in presence of the allosteric inhibitor will come and bind to the allosteric site. Once the allosteric inhibitor binds to allosteric site, a configuration change in active site will occur and the bound substrate will be released so that reaction cannot be forwarded, and the EI complex which is formed will be retained so that product cannot be produced. So coming to the examples of irreversible inhibitors, oxalic acid and citric acid inhibit blood clotting by forming complexes with calcium ions necessary for the enzyme metal ion activator. The best example for Irreversible inhibitors are oxalic acid and citric acid. The oxalic acid and citric acid inhibit blood clotting by forming complexes with calcium ions. So the inhibitors are calcium ions which are necessary for the enzyme metal ion activator. So this is about enzyme inhibitions, enzyme types of enzyme inhibitors and types of enzyme inhibitions. Thank you all.